is to get you thinking a little bit about how plants use sunlight for more than just photosynthesis. And that information is important to get first before we start getting into some of the plant communication ideas, especially when we start thinking of plants and their immediate neighbors. Because sunlight and, all the plants are, of course, trying to get to sunlight to do photosynthesis. So what happens when they're getting shaded out or so we're going to start looking at that idea with plant-plant communication and interactions. Then also today, there's a short little slide set I want to go over with you, just kind of something fun to think about. And then I want to go over the exam. So I finished grading the exam over the weekend. So, um, and there was one glitch where the answer key for one of the true falses was incorrect. And so I contacted the CTL. It looks like your grades got graded correctly, but they still have to work on getting the overall average recalculated. So, but I think your scores, it looks like they fixed that. So there's just the average I'm waiting to hear back on. So lecture on sunlight, little small thing I want to talk to you about. We'll go over the exam. And then those of you who haven't picked a paper yet for the Friday deadline, you have to hand that assignment in Friday, right? 10 o'clock. Those of you who haven't picked a paper, it'd be great if you could show me something before you leave. Some of you have already emailed me and I've given you feedback as of this morning. But since Friday's coming up fast, let's get that done today if we can. All right. If you've taken Plant Phys, this may look familiar. I know 3 of you are in Plant Phys. You'll get this again. So it'll be a nice review at that point. But let's get into how plants pay attention to light for things other than photosynthesis. All right. We all know by now their unique lifestyle of being rooted in place is very different from ours. And they have to deal with whatever they got dealt when that seed landed there and they started to germinate and grow. Mostly because they are rooted in place, that creates a situation where they have to be incredibly sensitive to their surroundings and being able to assess minute by minute what's going on around them and respond appropriately if they're going to survive and reproduce to get seeds put into the next generation. So they're constantly assessing signals from all kinds of different stimuli. When you think about how they're doing this in kind of the broad idea, they're assessing signals, and it's usually a case where a signal will get docked into a receptor protein of some sort somewhere, either on the cell membrane or in the cell. And then that signal has to get transduced into the cell so that the plant can make sense of what's going on and then respond appropriately. So this isn't unique to plants. All organisms are doing this. It's just that plants are really good at doing it and they're doing it at a much higher level than what we are doing. And so we'll think about this with regard to how are they dealing with sunlight? I may mention a few things about hormones as well as we work into this, because that's an important part of the transduction phase. So these are just some images to get you thinking about all the things plants are, or a variety of things that they're dealing with, whether it be, we've talked quite a bit about the fantastic movements of the Venus flytrap and the sundew. Has anyone noticed? I hope it's still there. There's a tree out at Presque Isle Park, the island. whoops, that does that. Has anyone noticed that? It's on the trail on the east side, kind of near the lookout where you can go out onto the lookout. It's kind of, I think it's beyond that a little bit. There's a tree that looks exactly like that. I didn't take that picture, but has anyone noticed that tree?
They're kind of just like hanging over a bit or
It's in the woods. I remember because when I was walking with my boys when they were younger, they were climbing all over it, and I got a picture of it. So it wasn't hanging over where they could have fallen over, but it was in the woods well enough. But um, close to the eastern part of the island. Anyways, I hope it's still there. I was told that that can happen intentionally, like people who, Native Americans who have lived in certain areas will do that intentionally to young trees as a marker, like a trail marker of some sort. And so I don't know if that is the case for the one out at the island or not. I know that maybe, well, another option might have been that there was something laying on top of it in that area of the trunk as it was growing, and it caused it to do that kind of bending around it. And then whatever was on top of it was um, taken away or moved away. Anyways, can you tell what might be going on here? Yikes. That's pretty amazing. It doesn't just tip over. So it's right along the coastline. It's getting constant buffeting, it looks like from the left in this case, of winds, really strong, probably laminar flow type wind moving really briskly across the tree at a pretty regular intervals, I'm guessing. And so they can create really fantastic additions to parts of their tree trunk to make sure they can really beef up certain aspects of it. It's called reaction wood, which they're trying to show here to make them stand relatively upright against all that wind force. So they have a lot of things to deal with, and signal transduction is their way of kind of making sense of it and then hopefully responding appropriately. I like this diagram. I think it's a nice flow chart essentially on how to think about this idea of signal transduction, whether it be from sunlight cues or whatever. So we have the environment or developmental signal, the receptor of some sort, and then often there is some kind of amplification. They often say second messengers play that role. They kind of amplify the signal. And then the transmission or transduction phase often involves hormones or electrical signaling as part of that process. And then you get the response, which ultimately typically is a change in gene expression to respond to that signal that they initially have received. And then usually what happens is at some point you'll need to, the fancy word is attenuate, signal attenuation, which means at some point you're going to have to like quiet that down. You can't always have that on all the time, typically. And so there needs to be a way to kind of quiet things down and say, okay, we're done with that, interpreting that signal. So with sunlight, we need to think about more of this idea of photoreceptors. We've mentioned this a little bit already when we looked at plant neurobiology and communication and behavior. So I'd like to focus on the photoreceptors that aren't really involved in photosynthesis per se and think about how else are they using sunlight. You can imagine as you hike through the woods, hopefully you did that this weekend where it was a little cooler. It's too hot in the sun. I don't like sweating all the time. Anyways, photoreceptors. In the woods, you think about all the shady and sunlit patches, all the different canopy levels of the plants. And so they're constantly dealing with this shade light variation. And when they are interpreting this, they're often using a suite of different photoreceptors. So what are photoreceptors? They are technically receptor molecules that can detect sunlight That's why they're called photoreceptors. Believe it or not, they go through this really kind of simple, elegant conformational change when they get exposed to light. And that is enough to set off a signal cascade of some sort into the cells and then some kind of response. And so because it's due to sunlight, they often refer to it as a photoresponse. So what they're doing is these receptor molecules absorb a photon of light and they're using that as a signal. They're not using it for photosynthesis. They're using it as a signal to do something, to shift their behavior. And when you look at these photoreceptors, they typically contain a protein portion and then a non-protein portion that's bonded to the protein. And it's the non-protein part that is absorbing the light. It's often referred to as a chromophore. It's in the notes if you want to see how to spell that. So the chromophore is the non-protein part. So when it does absorb that light, it actually shifts its shape. And that is enough of a process to allow for signal transduction to kick in. So it's not like they're expending a ton of ATP or doing anything super fancy. It's just absorbing that sunlight energy, shifting their shape, boom, that starts the signal cascade. They've been studying this for a long time. This is kind of the early stages when they started realizing that things are really dramatically different, depending on if the plant is grown in some kind of light or in the complete dark. So the plant on the right, poor looking things, they were grown in the dark, and that's how they ended up looking. Couple of things I think you can easily notice: different color, right? Really not so nice yellow color for leaves. I mean, leaves aren't supposed to be like that. So there's no chlorophyll. They were unable to get that done. So that's not good. And they're a lot taller. So the fancy word for this response is etiolation. And one of the big ideas here is that it's like, why would they bother to get so tall if they're in the dark? But from them, like I do this sometimes in a Plant Kingdom class where we just put some potted plants in the cupboard and shut the door and forget about them for a while. And then they'll look something like that. Well, they don't know they're in a cupboard. They think they're getting shaded out. So they're like, I'm getting shaded out. So they keep growing taller and taller because they think it's some nearby plant that's shading them. And if they just get tall enough, they'll finally hit sun. And of course, if you have them in the cupboard, they won't make it. But so this idea of growing really tall is just them sensing they're getting shaded and they're trying to deal with that and respond appropriately. The good news is if you take that pot on the right and put it under some light, it'll green up, its stems will thicken up, and it will behave like a more normal plant. It can survive. But it's definitely stressed out because, of course, plants want access to sunlight. So this was some of the first, like very casual things that they noticed about plants if you keep them in the light versus the dark. And then, of course, we talked already about Charles Darwin and his son Francis doing a ton of research on plants back in the 1800s and the idea of phototropism. And that took a while for everybody to figure out. It wasn't just the Darwins who did this, it was a bunch of researchers over time. And eventually they figured out not just sun streaming through the window per se, it's an actual particular quality of the sunlight energy. It's the blue quality, it's the blue wavelength of light that the plant is actually responding to, not the whole spectrum. So we know that when you put a light through a glass prism, it breaks into all the different beautiful colors. And so they did those experiments. They're like, okay, let's take some plants and just expose them to red light or orange light or yellow light. And they did that on and on and on. And they found that when they do unidirectional light for phototropism, the only one that got significant curving toward the light was blue. So that made them think, okay, it's very specific, blue light. So there's probably something, some photoreceptor in the plant that is picking up on that blue light. And then they went into the search for that. This upper picture is just, again, showing, how it absorbs most strongly in the blue quality of light means that that's what it's adapted to. That's the wavelength of light that it can preferentially maximally absorb. So they started figuring that out well after the Darwins were here and gone. And they said, okay, if you give unidirectional light, blue quality, it'll bend. So then they started hunting for photoreceptors. So when you look at the phototropism, the action spectrum, which means an action spectrum just means what's the plant doing essentially. And for phototropism, it's bending. And so they're looking at the curvature toward that light source. And again, you can see how its maximal curvage corresponds to the blue light. So again, this is just supporting the idea that it's blue light that the plant is listening to or paying attention to, I should say, seeing, not really listening, but seeing. And then they discovered a photoreceptor that they named phototropin. And they could see when you hit phototropin with a spectral scan of light, again, blue quality of light. So it has adapted to maximally absorb in the blue wavelength, which means that's its favorite wavelength. That's what's evolutionarily speaking, that's what it adapted to capture. And that's how it's being of use to the plant. So that was some of the earlier work going on with photoreceptors specific to paying attention to light. We talk a lot about the visible spectrum. Visible spectrum is just what our retina can pick up on. So it's very human-centric. It's 400 to 700 roughly. But be aware that other organisms, including plants, can pay attention to other wavelengths. And so bumblebees can see in the ultraviolet, for example. Plants can deal with other qualities of light besides the visible spectrum and use that quality for information, something that they need to pay attention to and respond to. Well, here's another idea of a different photoreceptor where they started these types of experiments many, many years ago. And these are lettuce seeds that are put onto wet paper towel or filter paper. And then they started playing with them. They knew that lettuce seeds typically need light. If you ever buy lettuce seed packets, it always says on the packet, don't bury in the soil, keep on the surface because it needs light to germinate. So they started playing around with this, with different qualities of light. And in this case, they're showing what happens to the seeds, whether or not they're going to germinate if you give them a hit of red light or if you give them a hit of far red light or keep them in the dark, what's going to happen? So again, this is just a narrow spectrum of visible light that they're looking at. So of course, in the dark, it's almost all of them just, they don't germinate. They're like, yeah, okay, we need light. You hit them with red light, okay, so just the red quality of light, most of them germinate. Then some scientists started to get smart and kind of like, hey, I wonder what will happen if we do this. And so in this case, they hit them with red light. And then right after that, they hit the plant with far red light. And you can see a very different response. Seeds didn't really germinate. So that got them going, ooh, what's going on here? So then they really started to get crazy. And you can see on the bottom, they said, let's just keep alternating red, far red, red. And basically, they came to the conclusion that whatever the plant saw last, so in this case, it was red, whatever it got hit with last, whatever quality of light it saw last, that's what it was paying attention to. So we saw that when it gets hit with red light, it germinates. So down here, even though it was red, far red, red, red was its last exposure, so it germinated. Whereas here, far red was its last exposure. And you can see how it is very similar to when it got far red up on the top there. Does that make sense? Okay. So this got them scratching their heads. So they were like, okay, there's some kind of pigment that maybe can pick up red and far red light and somehow it's able to tell the difference between those two qualities, and whatever it gets exposed to last, it has some different behavior patterns to that. So they started searching for this. I'm laughing because one of a student that went to NMU and got a botany degree many years ago, when he first came here, he grew up on a farm, a family farm, and I was assigned as his advisor. That was back when all the faculty did academic advising. And I said, well, what's your major? And he says, just general biology. And he knew I was botany person. And I said, oh, you don't want to go into botany? No, grew up on a farm. I had to do all these chores on a farm. I'm not doing anything with plants. 4 years later, he has a botany degree and did 4 years of research with me on plants. And then he goes to University of Wisconsin-Madison, and he's responsible for discovering the three-dimensional profile, like the architecture of phytochrome. So it's like, okay, from someone who went from, I don't want anything to do with plants, and he was responsible for one of the really important findings. So he did the 3D crystallography structure of the protein phytochrome. That's what he worked on for his PhD. So Dr. Jeremiah, he ended up in plants anyways. So never say never. All right, so these phytochrome pigments, that's what they were keying in on. They eventually named them phytochrome. It's a group of pigments. There's still a lot of work that they need to do on this. It's not like they know everything on this, but they see that it can toggle back and forth into these different shapes based on if it's getting red light or far red light. And it's okay, so I'm going to slow down on this because this is, at least the first time I learned it, it's kind of mind bending. So let's go through this carefully. So when we talk about phytochrome, we abbreviate it P sub R for the red form, which means, so P stands for phytochrome, phytochrome R. The subscript R is referring to red light. So when the phytochrome pigment is in the PR shape, okay, when it's in that shape in the plant, when it gets hit with red light, it can pay attention to that, and it will then change its shape. So P sub R just means it's the physical version of the pigment that absorbs red light really well. And as it's doing that, it changes its shape. When it changes its shape, it converts into what they call the P sub FR form. So some of you who had Plant Kingdom, this should sound familiar. So this is a good review. So P sub FR form is phytochrome far red. Okay. So this is just the version or shape of the phytochrome pigment that is fully capable of absorbing far red light. So if you have some of this pigment and you hit it with far red light, it can understand that and it starts to change its shape back to the P sub R form. So it keeps toggling back and forth, P sub R, P sub FR, depending on what quality of light. So P sub R means it's the phytochrome pigment that can really respond to red light. P sub FR means it can really respond to far red. And when I say respond, I mean change its shape back and forth, back and forth. It's kind of weird. So you have to imagine there's a bunch of these pigments in the plant. And of course, in the leaves, and if you think about a forested canopy, some of the leaves are going to be getting hit more with red light. Some other leaves might be getting hit with more far red light. So what you end up happening, if you think about the entire plant body, is that they'll have different amounts of these two types of phytochromes. And it's actually the ratio of how much P sub R do I have, how much P sub FR do I have, and those ratios are what's really important to the plant. They're keying in on that. and they're trying to figure out an appropriate behavioral response to that information. I think this visual is a nice way to think about it, especially back and forth between the two pigments there. All right, so here's the P sub R, the phytochrome R version. That is, so R means it's really good at absorbing red light. That's what it's paying attention to. So if you hit that pigment with red light, it converts it over, it shifts its form to the P sub FR form. Now, when it's in that shape, that pigment's like really good at absorbing far red light. So if you were to hit it with far red light, then it toggles back, changes its shape back to the P sub R form. Really weird. So they really haven't discovered another pigment that does this like this. This seems at least unique so far. I wouldn't be surprised if they find something else, but okay. So if, well, this slide, often they're thinking about this idea, and when they think about a biological response, often it has to do with flowering. So initially, when they discovered phytochromes, they were really interested in how does a plant know when it's time to flower? I think you know it just from living your life so far when you're out hiking around, some plants flower in the spring, some in midsummer, some don't flower to the fall. How are they understanding when to flower to complete their life cycle successfully, get seeds done, ready to go before winter, at least up here, they have to get ready for winter. So when they say biological response, it's often something like that they're thinking of. So what they mean is if a plant has a lot of P sub FR, which what that really means is it was exposed to a lot of red light, right? So if there's a lot of P sub FR in the tissue, the plant's going, ooh, I must have got hit with a lot of red light. Red light means sun. All right. So for the plant, if they're saying, I'm seeing a lot of red light, that means they're getting a lot of sunlight because sunlight has proportionally more red light than far red light coming in and hitting Earth and getting down to the point of leaves. So when you think about a canopy, a forested canopy with different layers, the very, very top of the canopy, those leaves, they're getting about the same amount of red, far red. But as you think deeper into the canopies, the understory, I like to think of it how the top trees that are getting full sunlight, we know that they like red light and blue light for photosynthesis. So if they're, I mean, one of their huge jobs of the day is I need to do photosynthesis. I'm going to absorb red light for photosynthesis. If they're busy absorbing a lot of red light, that means the understory won't get as much red light because it's been absorbed by the ones up above. But the understory can get more far red because the plants up top are like, I want red light, far red, not so much. I'll let that pass through and it can go down into the understory. Does that make sense? Okay. So as you think about a more natural ecosystem, you have to think about that in terms of layers of plants. And we do have to consider photosynthesis from the aspect that they really want red light because that's going to help with photosynthesis. But red light is also saying, oh, I'm really in full sun. This is great. If they're getting a lot of far red, that means I'm getting shaded. It seems in some manner I'm getting shaded. Okay. So let's look at this visually. This is the shape-shifting idea. So to me, the P sub R form kind of looks like an M, the letter M. I can't help but think about the YMCA dance because that looks like an M and the other one looks like a Y. So I think about YMCA. Am I dating myself or do you all know what that YMCA song? Okay. All right. So the PR, the so-called inactive form, which means it, when they say inactive, it means that it's the form, oh, boy. It's the form that if it's in the P sub R form, oh, this won't let me write on it anymore. And I have no idea why it's doing that. Laser pointer. Um, the P sub R form, that means the plant must have absorbed a lot of far red light. It must be getting exposed to that. And so it has a lot of P sub R in it. So let's look at that. So the P sub R here looks like the letter M. Here's that little non-protein chromophore that they're showing. Okay, so it's in this shape when it's absorbed far red light. And now we call it the P sub R form. The P sub R form is just really good at absorbing red light. So they're showing it, if it gets hit with red light, it shifts into the letter Y, the Y shape. And then we call that shape of the phytochrome the P sub FR form. And it's really good at absorbing far red light. And so if it does that, then we're back into the M form. And I'm okay if you describe it that on a quiz or exam, if you talk about the M or Y, P, to get the point across that it's shape shifting, that's fine. But understanding it's this change in shape between the two. And we give them different names depending on what type of light they prefer to absorb. But it's a little mind bending because you have to think about, okay, if the plant has a lot of P sub R, that must mean that the plant was getting exposed to a lot of far red light, which typically means they're being shaded out in some way. But if there's a lot of P sub FR form, that's saying the plant got hit with a lot of red light. And red light usually means sunlight. Great for photosynthesis and growth and flowering often. So when the plants decide to flower, they're actually paying attention to the ratio, how much P sub R, how much P sub FR. So plants are actually assessing how long am I in the light? How long am I in the dark? And so they can actually tell. And, circadian rhythms, they can tell when, like in the spring, when days get longer, they're picking up on that. And they're using that information to figure out when to flower. And then as summer goes by, days get shorter. Now we're into fall. Days are going to get really short. But these ratios of pigments are giving them that information. They're picking up on it and they know the appropriate time to go ahead and flower. They're paying attention to other things too. It's not just these ratios. They're looking at, especially in the winter, they have to pay attention to how many consecutive warm days are there. Like if we get a brief burst of nice weather in February, they usually won't do anything with that information. But if it's like 4 days in a row, then they'll start emerging out of dormancy, which could be disaster for us because February is not dormant, is not springtime up here. So it can cause a lot of problems with plants that experience winters. But anyway, so there's cold, warm periods they're paying attention to, wetness, light exposure, and of course the hormones shifting going on. They're paying attention to all that internally. Questions on the phytochrome shape shifting? If this is your first exposure to it, or even if you've had it in Plant Kingdom, please give this some thought because we need to be thinking quite a bit about these ratios as we cover how plants are assessing their environment, and especially when they start to grow in more natural ecosystems, or if you are just growing them even like in crop fields where they're planted really close to each other. They know if there's a neighbor next to them based on these ratios. And so we need to be thinking about these. This is just trying to get you to think about the understory idea, the many layers that you can see, and how that really does make a big difference to their phytochrome ratios, depending on if they happen to be an upper story or an understory. And they can have very different ratios. And that's very useful information for them. Here is just looking at sunlight energy striking. I think the blue line is the one striking the Earth. Then you get some of the absorption being shown for P sub R. You can see it's more in the red. Won't let me draw. You can see the peak is more in the red, whereas the P sub FR form, it's favored to absorb more in the far red frequencies or wavelengths. I think the first discovery about phytochromes really was in relationship to plant flowering. They were very, very interested in that idea. That's such an important pivotal step in the plant's life. But they've noticed that there's other things like the seeds, those lettuce seeds. They knew that that was something going on with red and far red light. This table is just to show that they have been making progress on what these phytochromes are doing for the plant. And different groups of the flowers you can see on the left. So there's a variety of different responses. So we'll get into some of this as we move into the topics of how plants are growing in different environments. So we'll be spending a lot of time thinking about phytochromes with some of their behavior responses. Another one though, with that blue light on phototropism, there's a couple of blue light receptors. And so here's another one, cryptochromes. So there's the phototropin we already mentioned, and then cryptochromes are another one. They're not just in plants. They are showing up in humans, mice, other organisms, bacteria. So it's not unique. They've been playing around with cryptochromes. They know that they're favored to absorb blue light. And it seems like it keeps their stems shorter when they get good doses of blue light. It seems to play a role in their circadian rhythms, being able to figure out light versus dark. It seems to play a role in their synthesis of this particular pigment called anthocyanin. And they also seem to overlap or play some roles along with phytochromes. So it seems like they often work in concert to help the plant assess what's going on and then respond appropriately. And solar tracking. So many plants are pretty good at solar tracking through the day. They can turn their leaves to face the sun the whole day long, no matter where the sun is in the sky. Okay. You're seeing a picture of the cryptochromes down low there, a mock-up of how they're changing shape with the exposure to blue light. So this is, oh, I forgot to show it in the other one too, or make a point of it in the other one. So when they say that the P sub FR form is the active form, what they're also referring to is it seems to be the major form that will interact with proteins in the nucleus to alter gene expression. So it seems like the P sub FR form plays around and interacts with transcription factors in the nucleus, which then will decide which genes get turned on or off. And so they're showing that again with the cryptochrome. They're showing that the active form here, and again, they're showing that that goes into the nucleus and it starts to interact with transcription factors or regulatory proteins. And that will shift the gene expression. And we already mentioned the phototropins as the ones that they figured out are responsible for that phototropism bending toward unidirectional light, but they've since discovered that they are really important for the plant in another way. It's with guard cell movement. And so when you think about the stomata on the leaf epidermis, how you have the two guard cells that are guarding the pore. And so how wide open the pore is, they know that the guard cells are paying attention to blue light. And so once the guard cells absorb, once the phototropin pigment in the guard cells are absorbing blue light, that's telling the plant, ooh, sunrise, it's time to open up and go ahead and start taking in CO2 for photosynthesis. So it's kind of the kickoff. If they don't get blue light, they won't open their stomata. Leaf movements, it's helping the leaf move to track the sun, the chloroplast movement. I'll show you, I think I have some pictures of the chloroplast movement. Yeah, I'll show you that in a minute. We already talked about phototropism. So really, it's the kickoff again with the idea of plants bending toward unidirectional light. It's the photoreceptor getting hit with blue light that then gets the auxin hormone sent over to the dark side of the stem and tells the dark side of the stem, grow, grow, grow. And that causes the bending over toward the light. And so phototropin has to get hit with blue light in order for all that to kick off. Once that happens, then auxin shifts to the dark side and then stimulates the dark side to grow. And that causes the bending. So here's phototropin, a mock-up of that, and they're trying to show once it gets hit with blue light, it changes its shape. But again, you don't see any input of ATP, nothing, really extravagant. It's just this very simple change in their shape, and that's enough to kick off this signal cascade, and ultimately gene expression patterns can change. So plants are using this phototropin protein or photoreceptor, sorry, to pay attention to how they should put their chloroplasts how they should arrange their chloroplasts in their cells to maximize photosynthesis, but not create any sunlight damage, because too much of anything can be bad. And so depending on what they're getting exposed to, if they're getting exposed to low light, they tend to form their chloroplasts in this behavior in the cell, where they'll have a layer on the top and on the bottom to maximize capturing sunlight energy. So under low light, they try to arrange their chloroplasts so that they can each chloroplast can get what it needs. Under highlight, though, you see they're almost hiding. Like the lead ones are getting full on highlight, but the ones behind it are kind of hiding behind the ones in front of them. And again, that's just a trick they do so that there's not any light damage caused to the chloroplast, because that would be pretty disastrous if the chloroplasts get all messed up with too much sunlight energy. It can start degrading DNA and membranes. It's just a mess. And then in darkness, they're not quite sure. It just seems like they almost go to sleep. You just kind of get toward the bottom of the cell. That's when they're looking at this. So amazingly, plants are even rearranging their chloroplasts. Okay. I think I'm going to stop there for now. There's still some more slides, but it's going to take a while to get into. And so let's take a break and come back at 3. And then after that, we will, I think I'm going to stop here until Wednesday for this stuff, but I wanted to talk to you about one other thing and then we'll go over the exam. So let's take a break.